It gives us great pleasure to welcome you all to this session on MDR TB drug trials in children and what the landscape is looking like at the moment. We've got 5 very interesting speakers to present over the next hour and a half. We would ask the speakers to keep to about 12 minutes, and that will give us plenty of time to have some questions after each one. So there's a timer at the front which will give you a countdown, and then we'll start jumping up and down. I would like to introduce my co-chair, Professor Simon Scarf, who works in South Africa at Tygerberg Children's Hospital. And my name's James Seddon. I'm a lecturer at Imperial College in London. So over to Simon.
Thank you. And so let me invite the first speaker, who is Dr. Tony Garcia-Prats from the Desmond Tutu TB Center. He's the director of the Pharmacokinetic Research Unit, and he's going to give the first talk.
Okay, good afternoon. Can you hear me okay? Okay. You said no? Maybe, can I hear you? Oh, there we go. Okay. Yeah, thanks for coming this afternoon to this session. There's really a lot happening in the MDR TB pediatric trial landscape, and multiple trials of delaminid, bedaquiline trials, and we're going to hear some emerging results this afternoon that I'm excited about. But what I want to spend some time on is a new study that's just been approved to go through to protocol development within the IMPACT network. And I'm presenting on behalf of a core group of people that's been developing this concept, myself, Annika Hesling, Suyeon Kim, James Seddon, and Simon Scarf. And the study is IMPACT 2020, shortened oral treatment for MDR-TB in children or SMART kids, a phase 3 randomized multi-center trial. So I did want to note our institution receives research support from Otsuka for implementation of the pediatric delaminid trials. So our rationale for a phase 3 trial in children with MDR is essentially threefold. There's a substantial global burden of MDR-TB. We don't know exact numbers, but modeling estimates are 25 to 30,000 incident MDR-TB cases in children each year. And these children desperately need improved treatment. So current regimens are long. Even the shortened regimen is at least 9 months. More importantly, they're toxic, and 20% of children treated long-term with injectables end up with some degree of hearing loss. These regimens are poorly tolerated by adults and children. But there's some different implications for children. So hearing loss, long-term hospitalization that are frequently found with current regimens occur during critical periods of neurodevelopment in children. That can have lifelong implications, can affect neurodevelopmental function later in life, psychosocial functioning, affects attachment. So the treatment regimens themselves are putting children at high risk. And even the new shortened regimen still contains an injectable for 4 months. What has been the approach to studying TB drugs in children to date? And this is an algorithm from the FDA. It's about extrapolating adult data for pediatric drug development programs, and I think it's helpful to go through it very quickly to understand the rationale. So the idea is essentially, if you have a drug that's shown to work in adults, what studies do you need to do in children? So the first, at the top of the algorithm, is the question, is it reasonable to assume that children, when compared to adults, have a similar disease progression and response to an intervention? So for TB, if you have a TB drug that works in children, it's reasonable to assume it will also work at least as well. Or if it works in adults, it will work at least as well in children. So then what studies we should be doing are PK studies in children aimed at achieving drug levels similar to those for adults in safety trials at a proper dose. This is the rationale for the existing delaminid and bedaquiline trials. Those studies are looking to find the optimal dose and the safety at that dose across the age spectrum. But are there instances where we may want to take a different approach? And truly, is the response similar? Actually, it may be that children respond better to treatment than adults. And if that's the case, they may actually benefit from shorter or less intense regimens. So if we answer no to that first question, then what we should be doing are safety and efficacy trials in children. So that's the rationale for this trial that I'll be talking about, SMART Kids, and for other studies like SHINE. And just to be clear, I'm not saying that we should be doing efficacy studies with each new TB drug in children. The existing approach is rational and useful, but there also may be instances where efficacy trials are needed and actually children will benefit from them. So our third point is that really there's a need for efficacy trials in children with MDR-TB. Children have paucibacillary TB, which tends to be less severe, and they would reasonably be expected to respond better to treatment than adults. And the data bears this out. MDR-TB treatment outcomes in adults in programmatic settings, roughly 50% are successfully treated. In those same programmatic settings, children with MDR have roughly 75 to 90% successful outcomes. So just to summarize the rationale, children may suffer disproportionately from existing treatment regimens, and at the same time would be expected to respond better than adults to shorter and less intense regimens. Additionally, the time really is right, so more children are being diagnosed with MDR-TB, and new and repurposed treatments are becoming available, including for children. So we assert that children with probable and confirmed MDR stand to substantially benefit from an efficacy trial. I'll share with you some of the design aspects, at least where it stands now, but there are a lot of challenges relative to adult trials that we've had to consider and build into the design of the study. And so a few of them are here. It's often not known at the time of diagnosis whether a child will have confirmed or probable disease, so children often present with clinical TB disease and an MDR exposure. In those cases of probable MDR-TB should be treated as MDR. It may be that some of those go on to have a confirmed diagnosis, others may not. For the same reasons, we may not always know the drug susceptibility pattern at the time of diagnosis, but we can't delay treatment until there's a confirmation of these aspects, or you can't delay entry into a trial. And some children will not have microbiologic endpoints, children who enter without a confirmed diagnosis. So we're proposing a randomized open-label 2-arm phase 3 non-inferiority efficacy trial, and we carefully considered some other designs, including some more complex designs like multi-arm, multi-stage adaptive designs, simpler designs like a phase 2 study, but ended up settling on more of a traditional approach with a non-inferiority trial. And one important aspect of the design is that we are planning to power this for the combined group of children who have probable and confirmed MDR-TB. This would really be representative of children treated for MDR in the field, and we want a result that's going to be generalizable. The inclusion criteria, so children 0 to less than 15 years of age, and we thought carefully as well about the upper age limit, and there are a couple of reasons we chose less than 15. We acknowledge that we're excluding older adolescents from this, but really the core rationale for this efficacy trial is that children have different disease and respond differently to treatment, so we want to include a population that has disease that's representative of childhood TB. Older adolescents will have more likely to have adult-type TB and respond more like adults. We also want to include the spectrum of MDR in children, so children with probable or confirmed pulmonary or extrapulmonary MDR or RIF monoresistant or pre-XDR or XDR would be eligible for the study, and HIV-infected and uninfected children would be included. The main exclusion criteria is probable or confirmed stage 2 or 3 TB meningitis or osteoarticular TB, and the rationale just being that drug penetration to these areas may be less good, and children with these forms of disease may respond less well. Where is the computer? There we go. Okay. So what are the arms? So children with MDR or RIF monoresistant will be randomized 1-to-1 to the control versus the intervention arms. I'll show you those in a second. Children with pre-XDR or XDR would not be randomized. They would be assigned to a treatment arm based on their resistance profile. So the proposed intervention arm in this study is a 2-month intensive phase with once daily delaminid, clofazimine, linezolid, levofloxacin, and pyrazinamide, and then a 4-month continuation phase with all of the same drugs but just a lower dose of linezolid. The control is the WHO recommended 9 to 11-month regimen. And then children who had fluoroquinolone susceptible pre-XDR TB, so injectable resistant pre-XDR TB, couldn't be randomized because they couldn't receive the control regimen, which contains an injectable, but they could receive the intervention regimen, so these children would not be randomized but would be assigned to the intervention regimen, analyzed separately. Children who had fluoroquinolone resistant pre-XDR or XDR TB would get a 6-month regimen with once daily delaminid, clofazimine, linezolid throughout the course, PZA, and PASS. A couple notes for the regimens. We're debating about whether to reduce the duration of linezolid in the intervention arm just to 2 months to reduce the duration-dependent toxicity and ensure that this is really a safe regimen, which we think we could probably do without sacrificing substantial efficacy. And then what is a notably absent medication here is bedaquiline. And we would love to include bedaquiline in regimens for children, but we don't have PK and safety data, and we can't reasonably expect to have PK and safety data across the age spectrum in the medium term, so we've been unable to include bedaquiline in these regimens. Just to give you sort of a visual for the study, so children who have probable or confirmed RIF-resistant TB would be eligible and can be enrolled. And depending on their drug susceptibility to quinolones and second-line injectables would depend how they were assigned. So if they're probable or confirmed susceptible to quinolones and injectables, they would be recruited and randomized to the intervention or control arm. If they have probable or confirmed resistance to quinolones or second-line injectables, then they would be assigned to a treatment arm, not randomized, based on the resistance pattern. And then there would be a small proportion of children who actually get randomized who are subsequently found to have resistance to quinolones or injectables who would then be moved to the pre-XDR or XDR group. The primary objectives are to compare the efficacy of a 6-month regimen versus the control regimen among children with probable or confirmed MDR, so it's really that group that's our focus. And a co-primary objective of safety and tolerability, again in the probable and confirmed group. Secondary objectives include characterizing the treatment outcomes and safety in the pre-XDR and XDR TB groups, and we've also built in some other secondary objectives around PK, acceptability, and cost effectiveness, and some other exploratory objectives around biomarkers and novel trial design. The primary endpoints, so successful treatment outcome, defined as either cure or probable cure without relapse at 52 weeks after initiating treatment, again in the probable or confirmed MDR group, and the safety endpoint, grade 3 or higher AEs, at least possibly related to TB treatment, again in the MDR group. And then also some secondary outcomes around treatment outcomes and safety in the pre-XDR, XDR TB group, and some others as well. We did some broad consultation around our sample size calculations, which can be tricky for non-inferiority studies. But 346 children, so 173 per arm, would be needed to demonstrate non-inferior efficacy of the intervention arm among children with probable or confirmed MDR TB with 90% power. So again, we're powering for that group. Our assumptions are there, so 12% non-inferiority margin. We're assuming 85% successful treatment in the control group, which is consistent with the small amount of data in the public domain about children responding to the short regimen. And then 87% successful outcomes with the intervention arm, 40% with a confirmed diagnosis, and this sample size includes 25% non-evaluable. So with a sample size of 346, we would be able to detect a reduction from 20% grade 3A adverse events to 8% with 80% power and 6% with 90% power. This is obviously a large study of children with MDR TB, and we had to carefully, and we have carefully considered feasibility as we've developed this concept. We do think there are sufficient sites to be able to implement a study like this, so there are existing sites that are experienced, have a burden of MDR, and experience with implementing trials. But there's also a lot of site development that's happening as we speak, so with the Phoenix Preventive Therapy trial, and also with Impact P1108, which is the bedaquiline study sponsored by Impact, and 2005, which is the delimited trial. So sites are developing within the network, and we believe there'll be good capacity actually to implement a study like this. We're estimating 30 months to complete enrollment, and that works out to 12 participants per month. So 346 sounds like a lot, but 12 participants per month in a multi-center study should be feasible. It will obviously depend on investment of resources and capacity, but it's possible. So what are the potential impacts of this trial? Obviously, it would depend on the outcome, but it could result, and we would hope it result in changed international guidance for MDR-TB treatment in children, and we've had some preliminary discussions with the WHO who've been supportive of the concept. The proposed trial, even aside from that primary outcome, will also do a lot, and we stand to learn a lot from this work. So understanding microbiological and clinical radiological treatment response in children with TB. We stand to learn a lot and gain a lot of experience with the novel and repurposed TB drugs, which really are the future of TB treatment. So even if the intervention regimen we're studying here is not ultimately the optimal regimen that goes forward, a decade from now, we will learn a lot from the safety of these drugs, optimal dosing of the medications included in this regimen that will inform their use in other combinations in children. The trial will build international capacity for pediatric TB trials, which we think will be needed in future with more trials needed. And also I think it's potentially exciting that the research could actually catalyze the diagnosis and treatment of children with MDR-TB, and we've seen this, that in routine care, few children are being diagnosed or treated for MDR, and actually the trials can build capacity and spur identification and treatment of children. We've seen that already. It is an ambitious trial, but we feel like it's ambitious for the right reasons, improving treatment for children with TB. So with the support of the TB community, pediatric TB community, I think this has a lot of potential. I would like to acknowledge Impact for their support of the concept and the protocol, and in addition to the core team, a number of people who have given really important input on the concept to date: Kelly Dooley, Steve Graham, Amita Gupta, and others. Thanks very much.
Okay, we have time for one or two questions. So, any questions from the audience? There, Jeff.
Thanks, James. Tony and I started discussing this about a week ago, but I don't know if the panel or Tony, you can talk a little bit more about the necessity of including PASS for that last regimen, because I think for those of us involved in drug development, seeing such an older, potentially more toxic drug being recycled for newer regimens is more unusual.
What would you suggest?
I mean, if the data for bedaquiline was there, but I guess is it even needed? I mean, do you feel that regimen that I don't remember the core components, but that it wouldn't be adequate with what you have?
So the question was really around the inclusion of PASS for children with fluoroquinolone resistance. Yeah, it's a fair question. I think there's a couple considerations that influence us. And you know, these regimens have evolved, and I think we'll continue to have some discussion before this is sort of a finalized protocol. But one, I mean, there is data that actually PASS can be an effective agent, so that's one. In our experience, it's been reasonably well tolerated in children, much better so than in adults. That's two. But there's certainly some limitations. I think if we had a choice of other agents, we would probably choose them. I think there's a concern that, how widespread, how available will PASS be in the future, given it's sort of not as such a high priority drug. So, I think there's some limitations to that. I think the major issue is there's just not a lot of other agents that could be used there, but I think we'll need to discuss that further, and it's a fair criticism or point.
Coming here today, I think you can hear me. I think this pointer seems to work a lot better than in the other rooms. I'm happy for that. I'm going to tell you the result on the bedaquiline crush study, that it was called, and the implications for dosing of bedaquiline in children. This was a bioavailability study where we compared actually not hold and crush, but hold and dissolve. And you see, I've even crushed the wrong word there. Sorry. Whole and dissolved tablets. I have no conflict of interest to disclose. So you all know about bedaquiline. It was the first new anti-TB drug with a novel mechanism of action coming to the market in more than 40 years. It's inhibiting the energy metabolism of the mycobacteria, and it has been shown to shorten time to sputum culture conversion and increase the rate of relapse-free cure in patients with MDR-TB when added to an optimized background regimen. It's also being developed primarily by TB Alliance for use in treatment of drug-sensitive TB, so this is potentially a very important drug that we want to have available also for treatment in children. And there is a pediatric formulation developed by Janssen, who's the sponsor, and this is currently in clinical trials. But the access to this formulation is unclear in the short and medium term. So we ask the question, would dissolving the standard formulation of bedaquiline taption be an option for dosing in children? And we tried to answer that question by performing a crossover study, whole versus dissolve, in adult healthy volunteers. And the focus of this study was to look at the relative bioavailability of the dissolved tablets, because the bioavailability is the absorption-related parameter that is also determining the overall exposure that is most likely to be affected by a difference in the formulation. And we applied a standard bioequivalence criteria where we wanted to look at, so assuming that there is no difference in bioavailability, we want to see that the 95% confidence interval of the dissolved compared to the whole tablets falls within a certain range, which is commonly set to 80 to 125%. And we also thought that in case we are not able to show that they are bioequivalent, we can use the information from this study and use the quantitative information about the difference, and with that suggest an alternative way of dosing the dissolved tablets. So that was a background. So in designing this kind of study, you need to consider that bedaquiline is a bit special. It has an extremely long terminal half-life. The estimate is 5.5 months or so. And previous drug-drug interaction studies that have been conducted in healthy volunteers are quite lengthy, usually around 6 weeks, and they have sampling over 14 days after each dose. And it's difficult to analyze this type of data with non-compartmental analysis, both because that even if you sample over 14 days, you will not capture the whole profile, and also because you will have a carryover between the first and the second dosing occasion. So you need to use model-based approaches to handle this type of data. But we wanted to design this study as smartly as possible, and look at what is the shortest sampling time and washout period that we can use and still be able to characterize a potential effect. For doing that, we performed clinical trial simulation using a previously developed population PK model of bedaquiline and the main metabolite, M2. And with this model, we simulated studies with hypothetical sampling scenarios starting from the previously conducted studies with 6-week period and kind of working our way down. And what we looked at was the power to fulfill this bioequivalence criteria. And we ended up with a study size of 24 participants, and we were able to reduce the number of PK samples after each dose from 17 to 11 and sample at 4 different days instead of 8 different days. And yes, this is the actual design. If I manage the pointer, yeah. So for the sequence was mixed. So half of the group started with getting a whole dose of bedaquiline. Bedaciline's 100 milligram tablets. So this is 400 milligram tablets together with water. We sampled them quite extensively over 24 hours. And then there was a washout period, and they got a second dose on day 15, which was dissolved, again sampling over 24 hours, and then they had to come back 2 weeks later to get a last sample, 366 hours after the second dose. So, yeah, in this way, you have both some patients that getting their first, or some participants that get their first dose as whole, and some that get it as crushed. So the carryover effect will be on both sides, so to say. And it went very well. This study was implemented at Task Applied Sciences in South Africa, and all 24 patients completed the study and the follow-up period. You can see we had a fairly young crowd with a median age of just over 20, a bit more than half of them were females, and the large majority were Black South Africans. In this study, we observed no QT prolongation with more than 450 milliseconds, and also no lactate values of more than 3 millimole per liter. And most interesting to me, we gathered a lot of nice concentration data, in total 552 samples each of bedaquiline, and the absolute large majority of these were detectable. And to the results. So the existing model that we had used also for the clinical trial simulation could describe the data very well. I did some smaller modifications in the parameterization, but we're quite quickly able to capture the data right well. And this is what I'm trying to show here, with the, as Paolo called it earlier today, this colorful plot. So we call it visual predictive check. So it's a way of both demonstrating the observed data and what the model is predicting that the data should look like. So up here in the corner you have the 12 patients that first got a whole dose of bedaquiline, and the black rings are the observed concentrations. The red solid line is the median, and the pink area is the 95% confidence interval of where the model predicts the median will be. So what we want to see is that these lines fall nicely within the shaded areas. And yeah, it's a little bit hard to see because the scale is over the 48 hours, but just eyeballing the data, I think you can already appreciate that there were no larger differences at all between the whole and the dissolved data. And fair enough, when we actually tested it formally in the model, we saw absolutely no difference in the bioavailability between the whole and the dissolved tablets. And the confidence interval of the relative bioavailability was 94 to 108, so that's falling nicely in the predefined bioequivalence criteria. So we could show the bioequivalence criteria were fulfilled. And what we did see, though, was that the mean absorption time was slightly longer for the dissolved tablets. So for some reason, to me a bit unclear what it could be, the absorption was a little bit slower. And if you look at what this would mean in terms of the typical profile of bedaquiline, it delays the peak with a little bit less than an hour and takes the Cmax down with 5%. But given the magnitude of these differences, and that we think that it's rather the overall exposure that will remain the same that determines the effect, we think that this has no clinical significance. So to sum up, we were able to implement a substantially reduced design to test the bioequivalence of dissolved bedaquiline compared to whole tablets. And bioavailability of dissolved bedaquiline was found bioequivalent to that of whole tablets. And this will now be used in the previously mentioned pediatric study of bedaquiline that is performed by IMPACT, P1108, where dissolved bedaquiline tablet will be an option if pediatric formulation is not available. So I want to acknowledge the volunteers and the staff that performed this study, and all the people in the protocol teams for the CRUSH study and P1108. Thank you.
So we have time for questions. Any comments, questions? Yeah, at the back. Sorry.
Hi, Kristen Cambili from the United States. I've read somewhere that bedaquiline is not very soluble in water. And when you say dissolved, do you actually mean in solution, or do you mean suspension?
Yeah, I think
Okay.
it's rather shaking it around until they don't see any particles, but it will, we haven't tested that it is actually dissolved, and I don't expect it to be, given that it's supposed to have very poor solubility.
Any other questions? Okay, thank you very much, Helen.
Hey, good afternoon, everyone. I'd just like to share some of our early experiences of the acceptability and palatability of a new and child-friendly formulation of levofloxacin. So the WHO in 2015 estimated just under 500,000 new cases of MDR-TB in children. And models indicate that this translates to more than 1 million child contacts exposed to MDR-TB each year. Young children, as we know, are at high risk of progressing to TB disease. Although treatment outcomes for children are generally good, MDR treatment is long, complex, expensive, and has serious adverse effects. And this means that the prevention of MDR-TB is a high priority. The fluoroquinolones are known to have good efficacy against MTB in vitro and are a core component of existing treatment regimens for children. They also have a relatively good safety profile, and this makes them good candidates for MDR prevention. The lack of child-friendly formulations, as other speakers have highlighted, is a real barrier to research and clinical care in MDR-TB. And the levofloxacin formulation in particular is very bitter and difficult to dose in young children. TB CHAMP is a phase 3 placebo-controlled double-blind trial to assess the efficacy and safety of levofloxacin preventative therapy in child contacts, and it's recently opened up in 3 different sites in South Africa. McLeod in India has developed a new and dispersible taste-masked 100 milligram formulation of levofloxacin for the TB CHAMP trial, and this work was part of a lead-in study for TB CHAMP. Before I continue, I'd like to share a somewhat personal story with you. A few years ago, the wonderful lady who works in my cousin's house was diagnosed with MDR-TB. And my cousin has had a 2-year-old and a 6-month-old at the time. And after consultation, it was decided to start MDR preventative therapy for her children. And I'd just like to share some of her verbatim reflections regarding that time in her life. So she said the tablets were extremely bitter. She was unable to hide the taste. And her children would then refuse any food that she'd previously tried to mask the taste. She said that cutting the tablets up was very difficult. She's an accountant, she wanted to get it exactly right, and she was always worried that she was giving too much or too little of the treatment to her children. She said she crushed the tablets, she tried to dissolve them, but they wouldn't dissolve in cold water, so she tried warm water, and then she was worried maybe she was inactivating the medicines. And lastly, she commented that the children often gagged and vomited the tablets with their breakfast, and then she'd have to repeat the dose. She needed someone to help her eventually to hold the children down to inject the medicine into their mouths. And she said eventually the children didn't want to eat anything that she gave them, because they didn't trust what mommy was giving them. So I think for me this just really highlighted the need for better formulations for children. So the aims and objectives of this study were really just to describe the acceptability and palatability of this new formulation, and to explore some of the factors that influence treatment adherence to levofloxacin in young children. Children who were included were all under 5 contacts of adult sputum-positive MDR index cases, and they lived in the household with the MDR case. Exclusion criteria was any TB disease at enrollment or children who'd been treated for TB in the last 12 months, any child who'd been on INH or fluoroquinolone for more than 4 months, and also concurrent exposure to an INH susceptible index case. So we dosed the children with 15 to 25 to 20 milligrams per kilogram of the 100 milligram formulation, and this was given by the caregivers once daily for between 7 and 14 days. And the caregivers could choose, they either gave the tablet whole or crushed, or they dissolved it or suspended it in water. And then we administered a Likert scale palatability and acceptability questionnaire to all the caregivers on the last day of their administration. We also had a team of postgraduate social science students who did in situ clinic and household observations and interviews with the patients and the caregivers. And they were asked about their experiences of giving this to their children, and then the data was collected and analyzed iteratively, so enabling unexpected findings to emerge. This is how the questionnaire looked. And that's basically what the new formulation looks like, and it dispersed very easily in as little as 2 mils of water. These were our first 3 patients enrolled in the study, and they happened to be triplets. And you can see a PhD social science student in the background just observing the dosing. So our results from the questionnaire: 27 children took part, 40% were girls, and the median age was 25 months, and all the children were HIV uninfected. At dosing, 81% of the children swallowed the entire dose or swallowed it with a small runlet. Others either refused it or spat it out. All caregivers reported that the tablet dissolved easily. They had no problem suspending it, and they were very happy with the drug volume that we used. 48% of the caregivers felt that their children either liked or really liked the taste of the tablet, and only 7 out of the 27 really disliked the taste. Almost all, 96% of the caregivers indicated that preparing the dose was really easy. 70% of the caregivers indicated that actually giving the dose to the child was either easy or very easy. And we did ask the caregivers then to compare this levofloxacin tablet with the previous medication that they'd been given. Most of them were already on preventative therapy, and 85% of them felt that the preparation of the new formulation was either better or much better than their previous, and 81% felt that the taste of the new formulation was either equivalent, better, or much better than the taste of the previous regimen. Results from the interviews: we noticed that the caregivers really changed the method of administration to ensure acceptability. And this was affected by the child's age and also the caregiver's prior experience of treatment, either their own or the child's. So they would adopt different strategies, not necessarily related to what we'd ask them to do, to facilitate treatment administration. And methods include hiding the treatment in food, praising the child, bribing the child, threatening the child, reducing the volume of water used, and even altering the number of pills and the frequency of administration. And they also used different reminders in order to help them give treatment. So they used cell phones, TV programs, some even sent the treatment to school with their child. Most of the caregivers commented that they were really relieved by the ease of administration of this formulation, and they were quite saddened by the fact that they needed to go back onto their previous adult formulation. Amongst the children, most of them were very young, but reactions varied from disgust to delight. One child spat it out immediately, and this little girl said that the pills are really tasty. So in conclusion, we found relatively good acceptability and palatability in these young children taking a new dispersible formulation of levofloxacin. We noted that caregivers adopted various methods to ease treatment administration, and they negotiated treatment manipulation in order to improve acceptability for their children. And really we feel that more child-friendly formulations will help children adhere better to TB preventative therapy in future. I have no conflicts of interest to disclose, and I'd just really like to thank the staff at the Desmond Tutu TB Centre, and the caregivers and all the little patients who took part, our funders, and Prof. Hesseling Saatchi Chair in Pediatric TB. Thank you.
Do we have any questions?
I'll ask an unfair question. Any idea when this formulation will be generally available?
I don't know about generally available, Prof, but we hope it will be available in the next couple of months as part of TB CHAM. And then we'll have more extensive data and acceptability data in particular, because all of the 1,500 children who will be enrolled will be part of an acceptability study. And after that, who knows, we'll have to ask McLeod.
Lindsay.
Sorry, thanks. I'll just say the levofloxacin dispersible tablet's actually available on the GDF catalog right now. So if your country procures through GDF, you can purchase it.
Thank you very much. One more? Not allowed to go just yet?
Excuse me, do you have any idea how is the cost of one treatment? The cost. How much do I have to pay for a pill? Thank you.
Is it on the GDF website? So I think the cost is on the GDF website, so you could be able to check the cost. I don't know off the top of our head. But it's pretty easy to find on the website. Okay, thank you very much, Sue.
Session is Professor Mwinga from Zambia. She's standing in for Dr. Musondo. So thank you very much for standing in at short notice.
Yeah, good afternoon, and as the Prof said, I'm standing in for Dr. Musonda, who works in my organization, Zambat. He was unable to travel. So we're going to change our direction a little now. I think we've had some good presentations on the clinical trials that are taking place in children, as well as the advances in the drug formulations. I'm going to spend a few minutes talking about the role of community engagement in MDR TB trials in children. And I think the last presentation was a real good example of the importance of thinking about how to engage with children and their communities as we set up these clinical trials within the communities. So the brief outline of the presentation is to talk briefly about the importance of community engagement, look at the importance of community engagement in pediatric trials, and then give a few examples of some of the work that has been done in both South Africa and Zambia in terms of engaging with children in terms of the clinical trials and the community engagement. So what is community engagement? I think community engagement has surfaced in the last few years, highlighting the importance of researchers not only just designing studies in their offices and then going out to the communities to implement these studies, but trying to ensure that communities are engaged in the whole process of the research study from the very beginning, if it's possible, so that there is ownership of these studies that take place. The community engagement is intended to ensure that there's protection of the interests of the communities, and the communities are actually able to contribute to the studies. And by so doing, it increases the likelihood that the communities will have ownership of the studies and that these studies will actually be beneficial to the communities. It also minimizes the potential for exploitation of individual participants and the entire communities by researchers. So why should we have community engagement when we are thinking about pediatric research? Generally speaking, children are a vulnerable population who require special protection. I think most of the studies that are submitted to ethical reviews boards, when there are children involved, they get special scrutiny. Community engagement is also important because as we have increased research in children, we need to engage the children as beneficiaries of the research so that the research can actually be relevant. Community engagement approaches are also required for the researchers to be able to inform, interact, and provide the relevant feedback from both the children as well as the adults that are involved. I'll now give a few lessons that have been learned by Dr. Musonda and his colleagues as they have looked at the whole aspect of community engagement. It is important to invest in general in community engagement. One of the reasons being is that in some of the communities in which these studies take place, there's relatively low exposure to clinical trials, and especially the science behind clinical trials. And so this affects the ability to get informed consent from parents and guardians of the potential research participants. And then when you look at the whole issue of MDR concepts related to either treatment or trials, these are complex and sometimes confusing to both the parents or guardians. Sometimes there's difficulty in understanding the difference between drug-susceptible and drug-resistant TB, and drug-resistant is often confused with disease potency. There's also some lack of understanding in the difference between TB infection and disease, and also what is the difference between active treatment and preventive treatment, the meaning of placebo, and things like that, as well as the care available to participants. And in some of these communities as well, therapeutic misconception is quite key, and parents or guardians may agree to take part in a study, not because they understand that it's a research, but maybe in the hope of getting treatment for their child. It is important to engage children as useful stakeholders. Children are not just there to obey commands. Children are little people. Children have ideas of their own. They form opinions, as we heard in the last presentation. There are occasions when a parent may want something for the child, but the child has different ideas. We have here an example from the Desmond Tutu Center, wherein a mother of a child that was participating in a TB trial had suggested to the child that she takes a medication during playtime in order to ensure adherence. The 5-year-old child refused that because she felt that if she had to take her medicine during playtime, it would become more obvious to her friends that she was actually on medication. So it is important to consult a child as even the regimens and the frequency of dosing is decided upon. Children can also be good stakeholders in terms of advancing the research agenda. In one study that was done in Zambia, in the ZAMSTA study, children were involved in an enhanced case-finding study. Key messages on HIV and the stigma were delivered to children at a level at which they could understand. And these children went home and found opportunities to talk to their parents and other household members about TB. Nearly all the children who were involved managed to have meaningful conversations at home and actually were able to help some of their relatives know about TB and actually seek care for TB. We have here an example of this study. Children, after going through the whole process of talking to them about TB, were asked to illustrate in drawings what they understood about TB. And here we have one drawing where the child drew a picture of a house, a patient lying on a table, and the other members of the family away. And basically for the child, this understood that if you have a TB patient, then it's because the people don't want to be near that patient because they may get the disease. In the next illustration, this child drew a picture of a mother and a daughter talking together in terms of providing information of how TB was transmitted from one person to another. So it is important as we think about community engagement to develop and nurture multiple community engagement spaces which are tailored to the needs of each of the stakeholders. So the way you approach a child will be different from the way you approach an adult. And to engage with these children and the communities at different spaces, for instance, engaging children in their natural environments, which would be at home, perhaps at school, perhaps in the playground. And also to use child-friendly ways of disseminating information about the studies. So children are basically vulnerable, and they may not comprehend the science and implications of participating in a trial. And it's important as one interacts with children and their parents to make sure they understand the difference between assent and consent. And for guardians to realize that their responsibility of protecting their child may actually infringe on the child's rights to make independent choices. So to be able to have a balance between protecting the child and recognizing the child's right to make decisions. And most of the children in these communities where studies take place are part of an extended family, and that sometimes also changes the dynamics as you discuss with the children. So basically, as we think about community engagement, we need to think about reaching out to the children within the context in which they are living, because they are affected by TB in different ways. So community engagement needs to address most of the fears that both the children and the parents and the guardians have about enrolling the children in the research, and ensure that we create interactive forums where there's an opportunity to discuss the various issues. So community engagement for pediatric trials requires appreciation of the social-cultural environment in which the children live and in which the children are raised and in which the studies will take place, in order to ensure that there's meaningful understanding of what the trial is as the children participate in these trials. So thank you very much.
Great. Do we have any questions about the role of community engagement or the way communities are engaged with trials for children, particularly drug-resistant trials?
Thank you very much. I'm Mari Tennyson from South Africa. And I don't have a question, it's a comment. I just want to thank you for emphasizing the extended family dynamics when it comes to community engagement. And, I sometimes think that we tend to underuse the word stakeholders. Because when you're talking about stakeholders, it covers a broad spectrum. And like you said, when you look at think of stakeholders in terms of family dynamics, we have the grandparents, we have caregivers, or children who are at school who are in the care of educators. So specifically identifying stakeholders gives their responsibility more meaning than just the word stakeholders.
I think that is very true, and especially within the African context, the nuclear family doesn't really exist in many areas, and they say it takes a village to raise a child. So when one is interacting with the communities, it's important to understand the power relationships, because a parent may want the child to participate, but if the grandmother or the in-law doesn't understand, the power dynamics may be such that you may not be able to get the participation. So ensuring that you actually involve everybody in the community to understand the reason why the study needs to take place, I think you get greater participation here.
Any other questions, thoughts, comments? Okay, thank you very much.